Walter did his PhD at the University of Pavia in Italy, moving to Bristol to work with Corny Cross in 1997. He then moved with Corny to the University of Sussex, where he won a Royal Society University Fellowship. He remained at Sussex for a couple of years before moving to the University of Sheffield in 2006 to establish his own research group. Walter's recent work has focused on the identification of spontaneous activity in inner hair cells, and he has proposed that this activity may indeed be influential in setting down the tonotopic map of the cochlea. I think it exemplifies, in terms of his strategy in his research, it exemplifies the debate that we've just had in regard from going from a reductionist through to a sort of studying an organ in physiology. Walter science is exemplified by a clarity in his writing and in his experimental designs. His papers published in Journal of Physiology, Journal of Neuroscience, Neuron, PNES recently, to name but a few. He really is an exceptionally talented physiologist, and it's a great delight to be able to ask him to deliver the 2011 Sharpey-Schafer lecture.
Well, first of all, thank you very much for the very kind introduction. I would like to say that for me it's a really great honor to be invited in this prize lecture. And I would like to first, before starting, to thank the organizer and the Physiological Society for giving me this great opportunity. So today, what I would like to talk about is about how the sensory cell of the mammalian cochlea, the hair cells, they are built basically, and how the initial development of this remarkable sensory cell is shaped by a combination of an intrinsic genetic input and also an electrical activity that is occurring in the cells spontaneously during immature stage of development, so before the onset of hearing. So the mammalian cochlea is present in the temporal bone, and together with the vestibular system that is important for the sense of balance, they form the inner ear. So what is the role of the mammalian cochlea? Why it's so fascinating as a sensory system? It's because the mammalian cochlea is able to detect and process a really wide range of sound frequency and intensity. And for example, humans, they are able to detect sound frequency from down to about 20 hertz to about 20,000 hertz. And at the same time, the mammalian cochlea is able to amplify the signal from more than 100 folds. So this is a remarkable properties. And this is not all, because the sensory cell of the mammalian cochlea, these hair cells, they can operate at a kilohertz rate. And also, their sensory mechanosensory organelle that I'm going to talk to you a little bit more later on, they can detect sound vibration in the order of less than, well, in the nanometer, but even less than a nanometer. And these properties are beyond the ability of any or most of the cell of the entire human body. So these cells are actually an amazing piece of biological engineering. Unfortunately, we have only 16,000 of these cells. Then these 16,000 sensory cells per year, they have to be responsible for the full sensitivity and dynamic range of the mammalian cochlea. And you are stuck with this cell, and you have to keep this cell for the rest of your life. Because unfortunately, the sensory cells of the mammalian cochlea do not regenerate. So if you lose them, because sound pollution, because genetic defects, or even aging, you lose forever. So you can't get this cell back. And obviously, what you develop is hearing loss or even deafness. Where are localized these sensory cells? The sensory cells are localized along the length of the mammalian cochlea. So if you cut the cross-section here, I keep touching that. You can highlight, you can see the sensory part is the organ of cortis. This is a scanning electron microscopy view, just that this is a diagram. So based on the stereocilia, or these mechanosensory organelles that I mentioned you early on, that are present on the top of this cell, you can distinguish one row of inner cells here and 3 rows of outer cells here. So the inner cells here and here are the primary sensory receptor. So are the cells that are important to send information to the brain. On the other hand, the outer cells are important for enhance the sensitivity and the frequency selectivity along the cochlea. And they do that because they are electromotile active. And this is thanks to a motor protein called named prestin. So they act as the amplifier within the cochlea. So the organ of corti here is present between 2 extracellular matrix. So here we have the tectorial membrane, and here we have the basilar membrane. There is another membrane here that is very important. It's called reticular lamina. It is important to divide basically this structure in 2 components. One on top, containing the stereociliary bundle, or this mechanosensory organelle, and the one on the bottom that is basically containing the basolateral membrane part. I'm going to talk a little bit more about this a little bit later on. So what is happening when you hear? So basically what is happening is that the vibration, oscillation in sound pressure within the ear canal are converted by the ossicles in the middle ear in a pressure, fluid pressure within the cochlear duct. So this pressure that is present in the cochlear duct will cause a vibration in the basilar membrane, and this vibration will lead to the displacement of this stereociliary bundle or organelle. When this stereociliary bundle deflects, they open very special channel, mechanoelectric transducer channel. And this is the first step in the sound transduction, where you have a conversion of a sound energy into an electrical signal. So this electrical signal now can be perceived by the brain. Because the properties of the organ of cortis, such as the height, stereocilia, the width and the thickness of the basilar membrane, but also the biophysical property of the sensory cell, they change along the length of the cochlea. So the cells are tuned on a different frequency depending where they are located. So what you have that the cells that respond best at low sound frequency, they are mainly localized towards the top, while cells respond best at high frequency, they are localized towards the bottom. And it's remarkable that this tonotopicity, this gradient, is maintained, is preserved throughout the sensory system up to the brain. Now, for the focus of this talk, I'm going to mainly concentrate on the inner cell development that are the primary sensory receptor of this organ. So, how an inner cell works? So, as I mentioned before, there is this reticular lamina that is separating basically this top part, the mechanosensory part of the cell, from the rest of the cell, from the basolateral membrane, so that we have 2 different solutions in the 2 sides. So the stereocilia bundle, they are immersed in a solution called endolymph, and this solution is high in potassium and low in calcium. However, if you look on the other side of the membrane, we have the basolateral membrane that is surrounded by a more classical extracellular solution. It's called perilymph, and it's a solution that is high in potassium and high in calcium. So what is happening is that during bundle displacement, a sound-induced bundle displacement of this stereocilia, what you have is that you move this bundle, and by doing that, you stretch elastic element called tipplings that connect adjacent stereocilia. When you stretch these links, what is happening is that you open mechanoelectric transducer channels that are thought to be localized on the top of the short stereocilia, as you can clearly see here. Now, when the channels are open, they let in a depolarizing current that is the depolarizing because it's mainly carried by potassium. And the driving force is actually an electrical drive, a large electrical driving force, about 150 millivolt. And this is between the potential on this side is about plus 90 millivolt, and the cell resting potential is about minus 60 millivolt. However, like most of the other properties of this cell, this large driving force is not there early on during the early stage in development. It has to develop. So before the onset of hearing, you have a very little or no mechanoelectric transducer current flowing through this bundle. However, when after the onset of hearing, this current is actually flowing through, is what is generating a receptor potential. And this receptor potential is important because it's driving the release of glutamate from the vesicle that are located towards the base of the cell, so that the signal can be passed on to the auditory afferent fiber. Now, as you can clearly see here, these are not conventional synapses, are called ribbon synapses, specialized synapses present in the cell, the auditory system, but also in other sensory systems, such as the vestibular and also in the retina. So what is happening, what is the role of this, of the ribbon synapses? Is to tether a lot of vesicle around this electrodense structure. They are called the ribbon. And the function of this ribbon is thought to facilitate the coordinated release of multiple vesicles. So I'm going to talk about the properties of the synaptic machinery a little bit later on in my presentation. So obviously this is a very complex structure. So the problem is that how we are going to build this structure. In most rodents, such as mouse, rat, and gerbils, the development of the cochlea occurs in about 3 weeks. And this is from terminal mitosis at embryonic day 14.5 to the onset of hearing that is postnatal day 12. So this is just an overview of the functional differentiation of inner cell and the outer cells over time. And at this point, embryonic 14.5 here, the cell fate has been decided. So it means that this cell will become air cells. However, they are not yet fully functional sensory receptor. They have to develop a lot of properties. And they do that pretty much immediately. However, during the embryonic period development here, inner cell and the outer cell, they are very similar, at least in terms of biophysical properties to each other. And they mainly differentiate to generate 2 distinct sensory receptors postnatally. So the postnatal development is where most of the properties are developing. And this is different, for example, to what is happening in humans. So in humans, the development of the cochlea is happening, everything is happening before birth, because the onset of sound is at about 5-6 months in utero. So how we study development in my lab, well, we study development of this sensory cell. Well, we use a lot of different type of approach, from cell biology, molecular biology, imaging, but mainly we do electrophysiological recording from sensory cells. They are localized in the mammalian cochlea. This is an example. So we don't dissect the cell, we don't dissociate the cell, but we work from an in situ preparation. So what we do, we dissect the whole cochlea, and we place at the bottom a recording chamber, and we fix with a nylon mesh attached to the metal ring. So then what we do, we place the chamber in a rotating stage of an upright microscope. And the reason why we use a rotating stage, most of this stuff we made because you can't find commercially. is because we study tonotopic gradients. So what we want to do is to be able to record biophysical properties of this cell located along within the same cochlea. And this is rotating stage allows us to do that. Also, we modify this rotating stage by having below the center here heating elements. So all our recording are performed at body temperature. So you see that there are a lot of different pipettes around. I'm just going through only the most important one. We have here the patch pipette that allows us to record current voltage responses. We have a thermocouple here in dark green that provides a feedback to the heating system, so it is ensuring that the temperature remains constant throughout the recordings. And then the last element I want to talk to you is this one in yellow here. This is a piezo-driven fluid jet. So with this equipment, what we can do with this item, we can create a stream of solution at the tip of the pipette. So if we put the tip of the pipette close to the stereociliary bundle, we can actually stimulate the bundle and mimic the in vivo situation. And I'm going to show a little bit later on about how we do it. So I'm going to talk now a little bit about the top part, this stereocilia, this sensory, mechanosensory organelle, how they look like, how they develop. This is a scanning electron microscopy from an adult inner cells. And you see that it's built with a row of stereocilia or decreasing height, so forming a staircase structure. However, this staircase structure is not there early on in development, during embryonic stage of development, but is mainly basically progressing and developing during postnatal stages. So one of our aim within the lab is to try to understand how the growth of this stereociliary bundle is regulated. And to do that, quite recently, we look at one particular protein, is an actin binding protein called EPS8. So EPS8 is able to have a different activity, both bundling and capping activity, and is implicated in axonal philopodia growth, but also in regulating the length of microvilli in the intestine. And we knew that both philopodia and microvilli are very, very similar from the structural point of view to stereocilia, in the sense that they are sustained by a core of polarized actin filaments that run along the length of the stereocilia with the barbed end towards the top. So our hypothesis was that the PS8 was actually a component of this stereociliary bundle. It was important for the functional maturation, for the growth and maintenance of this stereociliary bundle. So obviously we want to test that, but the first experiment we have done is actually testing whether EPS8 is actually important for hearing. So we did in vivo physiological experiment. And we did in vivo physiological experiment by measuring the auditory brainstem responses threshold. So the auditory brainstem responses are an indication of the activity of the auditory afferent fiber and therefore the linear cells. We look for the ABR threshold in control and in knockout animals, a PSA knockout animal. And what you find here, there is an increase in the ABR threshold. But this increase was very high. It's outside the normal sound stimulation, which is completely unphysiological. So these mice, in reality, they are completely deaf. And this, for us, was a clear indication that the PS8 is actually important for hearing. So we went on and looked at the localization of this protein, EPS8. These are immunolabeling experiments, and these pictures are obtained with a confocal microscopy, and the plane of focus here at the level of the stereociliary bundle, so on top. We labeled the stereocilia with F-actin in red, and EPS8 is in green. And you see that EPS8 is labeling the tip of the longer and the shorter stereocilia. So definitely, we know now that EPS8 is important, is present, is expressed in the stereociliary bundle. So we want to see whether it was actually crucial for their development. So we did a few more experiments with scanning electron microscopy of adult inner cells. This is a typical staircase structure of adult inner cells. Then when you look at the knockout, you see quite a few differences. The first one is that you have extra raw stereocilia. And the second one is that the stereocilia are much shorter. And it's more clear, more evident with transmission electron microscopy. This is the same scale. You see that all the stereocilia were shorter, but especially the longer stereocilia was reduced about 70%. So we did many other experiments, and we basically conclude that EPS8 is actually essential for the growth and maintenance of this stereociliary bundle. Actually, the effect of the EPS8 is more complicated than that, because EPS8 is also involved in the functional maturation of the basolateral membrane part of the cells. But I'm going to talk to you more about this part a little bit later on. So we found that EPS8, at least this protein, is involved in the functional maturation of the morphology of this stereocilia bundle. So what is happening to the transducer channel? Is the transducer channel there early on in development? Is actually functional? And in order to do that, we can do this easily in vitro by using the element that I described you early on, this piezo-driven fluid jet. However, I would like to remind you that in vivo, this bundle will be stimulated by sound only after postnatal day 12, that is the onset of hearing. However, there is nothing that prevents us from testing what is happening to this channel before the onset of hearing. If we do this with the fluid jet, as I mentioned before, this is the tip at the end of the pipette, and we can either blow or suck solution inside of this pipette, and therefore we can stimulate the bundle, and we can record the mechanoelectric transducer current using a patch pipette attached to the cell body. And this is just out of focus, is the fluid jet. So because of the transducer channel is partially open at rest, I mentioned before there is a resting tension in this bundle that is keeping the channel partially open. We can modulate the channel both towards inhibitory and excitatory direction. So what is happening? So if we move towards inhibitory direction, so we suck solution into the fluid jet, so we move the bundle towards the shortest stereocilia, we release the resting tension, we close the channel, and in fact here you see that we reduce the holding current and we reveal the smaller transducer current is available at rest. If we move the bundle towards the excitatory direction, we open the channel further and you see a larger transducer current. And so we apply this technique to different stages of development, and you can see that during embryonic stage of development, we have very low or little transducer current when we get close to postnatal day 0. But mainly the transducer current is developing with postnatal development. It's increasing linear in terms of sites and also in terms of resting transducer current. This is actually very important because it is, at least from a physiological point of view, because it's involved in setting the resting membrane potential of the inner cell, and therefore the resting neurotransmitter release. So we have basically the sensory part of the hair cells that with time they show a linear progression in their morphological and biophysical development in order to generate a mechanosensory organelles that are able to respond to nanometer displacement. So it has to be set very carefully in this case it takes about a couple of weeks or so. But while this part is developing, what is happening to the rest of the cell? Are the cells waiting for sound to stimulate the cell, or they are actually active? And actually the cells, they are active because they fire action potential activity before the onset of hearing. They are spontaneous action potential activity. This is a continuous recording from a postnatal day 3 inner cells. Why we are so interested about this action potential activity is because this early electrical activity has been shown well before we started to record this in other systems, that is crucial for the development of ion channel, a reorganization of synaptic connection within the CNS, also within the retina. So we think a similar type of a role could be also present in the inner cells. Action potential activity, however, is not present throughout immature stage of development. Not all pre-hearing cells, they fire action potential. So if we look just post-mitotic cells, so embryonic day 15, 16, these are the voltage responses. So they are large voltage responses, both towards the negative or positive to the resting membrane potential. And this because they only express two channels. One carried a classical delay rectifier, and one carried an inward rectifier. However, from around the day before birth up to at least the beginning of the second postnatal week, inner cell they fire action potentials. And you see here there are many more ion channels. Basically, we get the upregulation of a sodium, a calcium, and a small conductance calcium-activated potassium current. Unfortunately, the action potential activity, this action potential, they are too slow to be able to follow continuously high-frequency signal. They are characteristic of mature sensory receptor. So the cell actually, they have to change again. And they do that in a matter of few days at around the onset of hearing. And what is happening is that now the voltage response, they look like small engraded receptor potential and sustained receptor potential. And this is because we have on one side the downregulation of immature type current. These are only expressed before the onset of hearing. And on the other side, we have the expression of two new current that are characteristic of adult cells. One is a BK channel. This is carry a very fast activating calcium-activated, large conductant calcium-activated potassium current. And the other channel is the KCNQ4 that is carried is a very negative activating delay rectifier. And the combination of these changes of the downregulation of this channel, upregulation of this one, you change the voltage response from action potential to non-action potential. This might be surprising, quite surprising for some of you. The action potential of inner cells, they're not driven by sodium, but they're driven by calcium. So these are all cell recording from immature postnatal 3 or P4. And you see that when we remove calcium, this action potential, they completely disappear. But then, if you reinstate calcium, in this case we use 1.3 millimolar, they start again. That is a physiological concentration. Now, these are spontaneous activity. Obviously, we want to be sure that these cells, they were really spontaneous active, and this was not some kind of artificial effect of the fact we are in all cell configuration. So we have a continuity between the patch pipette and the cell. So we verify that by using another method. This is much more physiological. It means extracellular recordings. So in this case, the patch pipette is attached to the cell, but we don't break the membrane, so the cell remains intact, and especially we don't interfere with the calcium buffering system that actually is crucial for this cell. And when we do that, we can still record spontaneous activity that in this case is in the form of inward capacitative transient. When we remove calcium, even in this condition, the activity is abolished, and then is starting again when calcium is replaced. Now, one of our interests was to find out whether this action potential activity we knew is present was different along the cochlea. And what we found is that if we were looking at the top part, the low frequency part of the mammalian cochlea, the action potential are in the form of a very irregular action potential activity, and much more irregular that if we look for in cell, they are localized towards the base. And this is also very evident if we plot the interspike interval as a function of time. The spread of these dots indicates a much more irregular activity than the one that we have in the base. So when we found that actually there is a difference along the cochlea, we were actually interested in saying, well, what is actually determining this difference? Is there some interest in mechanism that is responsible? We found actually that the different pattern of activity was largely dependent on the activity of the inhibitory efferent system. So what is the efferent system? So this is the efferent nerve terminals that are contacting the inner cells only during pre-hearing stage of development. Then there is a restructuring of the fiber around the cell, so that in the adult animals, efferent system, they only activate the auditory afferent fiber. This efferent system represents a feedback to the cells, and is an inhibitory feedback because acetylcholine that is released by this terminal is able to activate alpha-9, alpha-10 nicotinic receptor, which are highly permeable to calcium. However, because very colocalized, there are potassium channels. This calcium will activate these SK2 channels, and the final effect is to hyperpolarize the cell. So that's the reason why we have an inhibitory effect. And what we found that the activity of this efferent system is much more pronounced at the apical part than in the basal. And because of that, the cell localized in the apex, they are kept to a resting membrane potential that is slightly more hyperpolarized than the resting membrane potential of the cell present in the basal. And we found that this is actually sufficient to make the pattern or action potential activity in the apical cell more irregular. Now, here I want to just remind you that the major functional input of pre-hearing cells is this action potential activity and not the transducer current. So our hypothesis is that this gradient in the pattern of action potential activity along the cochlea could instruct or control the tonotopic differentiation of these inner cells during development. And in particular, one of such control, what we believe could be one such control, could be on the functional maturation of this other connection, so the auditory afferent fiber with the presynaptic machinery. So why the synaptic machinery has to change during development? The main reason is because the synaptic machinery has to be able to encode with accuracy the different receptor potential that these cells are showing. In this case, pre-hearing cells action potential activity that is similar to a digital signal, and in adult we have graded and sustained receptor potential. This is more similar to an analog signal. And these two different, the information encoded in these two different receptor potential have to be transmitted with high fidelity to the auditory afferent fiber. Well, it's clearly why you want that in adult stages, because you want to hear properly. But why is important also during pre-hearing stage of development is because we believe that this information are important for reorganization of synaptic connection. So in both cases, the release of neurotransmitter is a calcium-dependent process and can be measured electrophysiologically using cell membrane capacitance. Now, I'm sure there are a lot of experts about this in the audience, but I just want to go through the basic principle of this type of measurement for anybody who's not so familiar. Basically, what is happening is that the receptor potential will depolarize the cell. Cell depolarization causes the opening of the calcium channels. So calcium will flow into the cell, and what is happening is calcium will trigger the fusion of vesicles that are localized near to the membrane. When these vesicles are fused, they release the contents in the synaptic cleft. but in the same time, they incorporate the membrane, their membrane, into the membrane of the cell. So basically what is happening is these cells, they are growing. They is linearly with the number of vesicles. So if we know the surface area of one vesicle, and we know actually how much the cell has grown, then we can calculate or have an estimate of the number of vesicles that be infused with the membrane. And this is one of such experiments. So on top is the protocol that we use. The cells are kept at a resting membrane at the holding potential about minus 80 millivolt. And then we apply a depolarizing voltage step. In this case, I just show one for clarity, that is a minus 20. That is usually the peak for the calcium current. So what is happening is that we record simultaneously the inward calcium current and the capacitance, and the cell capacitance. If you look before and after the stimulus that is coincides with the calcium current, you see that there is a change of about 30 femtofarad. And 30 femtofarad correspond on average about 600, 700 vesicles being fused with the membrane. Now, if we plot the size of the calcium current at different voltage steps and the corresponding change in membrane capacitance, we get what we call a transfer function. So is an indication of the calcium dependence of the process. So in a very simple term, means that in this case, we have a high order calcium dependence with a power of 4. So this is the fit with a power function. And the power 4 means that in very simple terms that you need about 4 calcium ions to get a release event. So why this calcium dependence is actually ideal, let's say, to encode this type of signal? It's because when you have an action potentials, if you want to keep at the postsynaptic all the information that are encoded in a different pattern of frequency along the length of the cochlea, you need to make sure that the signal that you are going to transfer mainly is occurring at the peak of the action potentials. So basically, you want a maximal change in membrane capacitance at the peak of the action potential and very little in between. Otherwise, you lose information that are determined by the pattern and the frequency. And this is exactly what this power function is ideal, because you see that the maximal change in membrane capacitance you have with the maximal current or maximal stimulus, so at the peak of an action potential, and very little or nothing in between. But however, this is actually not ideal for a dull cell. So what the synaptic machinery has to do to be able to encode this signal accurately? And what is happening is that they change the calcium dependence. They change from high order to linear. So in this case, the system is efficient not only for large stimuli, but also for very small stimuli. And this is what you need if you want to encode not only large stimuli, but also very small stimuli. So the machinery has to develop in order to be able to encode different receptor potential. How this development is occurring? At least in the high frequency region, so for cells that are respond best to 1 and the kilohertz, so bigger than 1 kilohertz, we have that the power function that is dictated by the fit change from about 4 to linearity. And this occurs quite rapidly, a few days just before the onset of hearing. However, if you look at the low frequency cells, so in the hertz range, a few hundred hertz, what is happening is that the calcium dependence remains so high order power from about 4 to about 3. Why is that? Again, it's related to the type of receptor potential. So I already described you the action potential and the sustained receptor potential. But low frequency inner cell in adult animal, they show this large AC component. They are dominated by this AC component that in some respect is more similar to the action potential activity. So again, the power function will be more suited to encode this type of activity. So when we were working on this change in change in the power function, we were really query about whether single molecules could be responsible for this change from a high order to linearity. And we know that the classical calcium sensor of neurotransmitter release are synaptotagmins. In most of the classical synapses, what you have is that either, in most cases, either synaptotamin 1 or 2 that are important for setting the calcium dependence of the machinery. And generally, what they have, they produce high order calcium dependence. So we actually don't know, or let's say that it's still not clear whether synaptotamin 1 and 2 are expressed in the mammalian cochlea. However, even if they are expressed, they can be responsible for the linearization because they determine high order calcium dependence. So basically, we were looking through different synaptotagmins. And one that is quite unconventional is synaptotagmin 4. Because different from most of the other synaptotagmin, doesn't bind calcium to the calcium sensing domain. So this synaptotagmin 4 could have the role of reducing the overall calcium dependence. So we decide to look into that. And first of all, we look at immunolabeling, whether we could detect synaptotamin 4 within the cochlea. So we look at immature stages and also in low frequency inner cells from adult animals. All of this, they show high order calcium dependence, and we couldn't see any labeling within the cells. However, when we look at a high frequency inner cell of adult animal, that they show linear calcium dependence, synaptotamin 4 was present and colocalized partially with calcium channel at the presynaptic site. So this was a very good indication that synaptotamin 4 could be actually important for the linearization. We had to test that, and we did that using synaptotamin 4 knockout mice. So these are recordings, membrane capacitance recordings similar to the one I showed you before from adult inner cells. And you can see that the size of the calcium current and that of the capacitance, the cell capacitance, was not actually different between the control and the knockout. However, when we plot the calcium dependence, so the relation between calcium current and the change in membrane capacitance, what we found that the knockout did not develop the linearity. So they remain completely immature. So they still show high order calcium dependence. So this clearly points that the linearity in the calcium dependence is regulated, is determined by synaptotagmin 4. Now we know the molecules, we still don't know the trigger. So what is actually important for triggering the expression, the linearization or the expression of synaptotamin 4? And my hypothesis that I mentioned you before is actually this action potential activity could be actually the trigger for this linearization. How we could test that? One is, for example, to have an organotypic culture, modified the action potential activity in organotypic culture and see how the synaptic machine is developing. Unfortunately, the mammalian cochlea does not develop in culture, so it stays forever in a mature configuration, possibly because you don't have the efferent system that is feeding back to the cell, probably for other reasons, we don't know yet, but the mammalian cochlea does not develop in vitro. So the only other way that we thought is basically using a mutant mouse that has an altered action potential activity as a constitutive activity. So it's because of some mutation, the action potential activity of this cell is altered. And before we go into that, I want to show you how the action potential is, what are the currents that determine the action potential. So I mentioned that a calcium action potential in inner cells are mainly calcium dependent. If you remove calcium, you don't have action potential. However, you can see here you have many more other channels that contribute to the shape. One current that is actually quite important is this one. It's a small conductance calcium activated potassium current. And this is important because it's able to make the repolarization phase of the action potential quite robust. If we block this current with a specific blocker apamine, what you have is that the width of the action potential, they get much wider. So basically what you have is that both the pattern and the frequency of this action potential is altered. So we decide, well, this could be the right model. So we start to collaborate with John Adelman in the States on the SK2 knockout mouse. And this is an action potential activity in all cell configuration from an immature postnatal day 4 and show a normal activity. Then when we look at the knockout, we found that the cell is able to respond to have action potential activity. However, with time, you get a gradual depolarization until the cell they reach a steady depolarization. They can stay there for some time, then are able to repolarize again, fire action potential again, and do that in a cycle basis. So this, in theory, could be a right model. However, we wanted to be sure that this behavior was due only to the lack of the SK2 channel, because sometimes you get indirect effect due to the fact that these mice, they've been developing for quite some time without the channel. So we decided to test that in control animals. And we can do that because we can mimic the knockout animals by artificially blocking the SK2 channels with apamine. And in control animals, when we apply apamine throughout the recording, you see that we get exactly the same behavior. So at this point, we were actually confident that this was the right model. So we have a model with an altered action potential activity, so we can go and have a look at whether there was any defects in the maturation of the synaptic machinery. We look again adult inner cells, capacitor measurements. This is, I'm going to show directly the calcium dependence of the neurotransmitter release. And in control animal, everything was normal, as expected, linear calcium dependence. Then when we look at the knockout, we see an effect that was very similar to that of the absence of synaptothamine 4. So the synaptic machinery remains completely immature. So it's not able to develop, and we link that to the fact that the action potential during mature stage of development is not normal. I want to point it out that the SK2 channels, they are not expressed during adult stages. So any effect they have during mature stages of development. So the next question is action potential activity sufficient for the functional maturation of the synaptic machinery? And the question is no. And the simple reason because you need actually a genetic input. And this came from very recent experiment that we done on the EPSA knockout mouse. This is the mouse that I've been showing you early on at the beginning of my talk. That I mentioned that in addition to be important for the maturation, for the growth of stereociliary bundle, is also important for the maturation of some of the properties of the basolateral membrane. And one of these one is the calcium dependence. In the absence of EPS8, this actin binding protein, what we have is that the synaptic machinery remains immature. And this, despite the fact that they have completely normal action potential activity. So basically, in order to, basically the functional maturation of inner cells at around the onset of hearing require, first of all, an upstream genetic input so that the action potential activity can exert a role in the tuning, in the refinement of the functional maturation of inner cells, most likely on the expression of synaptothermin 4. However, all these changes occur quite far into the development of these inner cells. So we had another idea, another hypothesis that basically we thought maybe we have another element, another molecules much early on in the development that is important to regulate all the elements you need to mature for inner cell to mature. And actually, even better, maybe you have actually a master regulator that is acting at this level and is responsible for coordinating the functional maturation of both sensory cells, inner cells, and the outer cells. So, which could be the possible candidate? Which are the possible candidates? Well, we thought transcription factor. Another possible candidate are the microRNAs. And actually, we were kind of intrigued about the microRNA because microRNA are small non-coding RNAs that are able to control, to regulate a wide range of protein-coding genes. They are implicated in the development, in the cell differentiation, and actually are crucial because they are important for a lot of diseases. So we had the opportunity to look at the only mouse model at the moment for hearing loss with a mutation for one of these microRNA. And that is, the mouse is called diminuendo. It's a mouse that was derived through the ENU mutagenesis program. And this mouse has a single base change in the seed region of microRNA 96. So this is a very conserved region among species. And microRNA 96 is very specific to sensory organs. And within the cochlea is expressed in the hair cells. And as I already mentioned a few minutes ago, it's important because it's causing progressive hearing loss in both mice and humans. So it's a very good model also for hearing loss. So we said, well, we have a mutant mouse with a mutation in microRNA 96. Well, we decide to look at the functional maturation of the sensory cell. And we look at a lot of different properties. I'm going to just summarize the major finding. We look at morphological maturation. These cells, they usually grow in size, so this is morphology. We look at another morphological feature, such as the reorganization of synaptic connection. We look also at biophysical properties, such as ion channel change during development, the functional maturation of synaptic machinery. And what we found that in this mutant, none of these properties actually was able to reach maturity. Not only for the inner cells, but also inner cells, they were affected at the same level. And actually was quite a surprise when we got the results, because this was completely different from any other mutant mouse or knockout mouse that I looked before, because the development of both sensory cells, basically this cell, they were stuck at a stage of development that was pretty much around postnatal day 0, around this age. So the absence of microRNA 96 was causing a coordinated break, all the morphological, physiological, functional maturation of both inner cells. So it was actually able to act as a master switch for the functional maturation of the entire cochlea. So I would like to just summarize and say that building a mammalian cochlear cell is actually a quite complicated task, and requires a high level of coordination between an intrinsic genetic program, and this genetic program has to be in place before the action potential activity, this intrinsic activity, can actually exert a role of refining the system at around the onset of hearing. Now, in the last, let's say, 10 or 15 years, the hearing research has made really a big progress in the understanding of our how sensory cells are constructed, are built. And this has been driven quite substantially by the study of genes that have been involved in hearing loss. Despite this effort, unfortunately, we are still very far from, let's say, the comprehensive understanding of all the elements that are actually required for building, for the R cells to develop. For example, we still don't know We still don't know the nature of the mechanoelectric transducer channel. That is the first step in sound transduction. So you need this channel not to convert sound energy into an electrical signal. We still don't know the nature of this channel, despite the fact there are huge amount of big labs around the world trying to find this. We also don't know a lot of elements and mechanisms responsible for the release of neurotransmitter from these specialized ribbon synapses. Also, we don't know, for example, how this different pattern of action potential activity along the cochlea is able to influence the maturation of the sensory cell. And these are only very few of many questions that still we need to answer. So I'm sure that we are going to have a very exciting and very challenging time ahead. And I guess mainly because these cells, at least from my point of view, they are getting really more complicated every day. And I would like to obviously thank all the people, previous and current collaborators that contributed to the work I've been showing you so far. It's not very clear, but especially at the University of Sheffield, my lab, Stuart Johnson, Stephanie Kuhn, and Tobias Ekrich that are there in the audience that performed most of the electrophysiological experiment. At Sheffield also, Matthew Holley and Carol Lackney. And then my collaborator in the UK, and especially at Keele University, David Furness, and Karen Steele at the Sanger Institute. And then my collaborator in the in Europe, and especially the laboratory of Marlies Knipper at the University of Tübingen in Germany. And then my collaborator in the States, and in particular Robert Fattyplace for recent work on mechanoelectrical transduction. Then I want to thank my two major sponsors, the Physiological Society, the Royal Society, for supporting my fellowship, and also the Wellcome Trust that is supporting the majority of my research. And also I would like to thank Action on Hearing Loss, the former RNAD, and Deaf Research UK for their long-term support in project and equipment grant. And more recently, Westfield Health and the Physiological Society for equipment and travel money. Then I would like to thank the Department of, it's not physiology, but it's the Department of Biomedical Science at the University of Sheffield for the great support and the fabulous environment, very friendly environment. And then I would like to thank few people that for different reasons are very special to me. Professor Matthew Holley at the University of Sheffield for being so supportive during the last few years. My senior postdoc, Stuart Johnson, for his hard work during the time I've been in Sheffield, setting up my lab. And Stuart is a really talented young scientist. I'm so happy to say that has just been awarded with a Royal Society University Research Fellowship starting in October this year. And finally, my family, my 2-year-old daughter and my wife for being so patient throughout all this time. And the last things, I don't want you to get confused. This is not Sheffield. That is why I put, that's actually where I come from, and it's Italy. Thank you very much for listening.